Hi everybody, welcome back to the next video. So before we move to the next experiment that we want to discuss, I just want to highlight a couple of aspects associated with the previous experiment. We ended the last video with the discussion of this experiment that the authors did, where the concentration of the antibiotic on the plate transitioned from 0 to 1000 times MIC in one step. And the results that they report is that obviously bacteria quickly expanded to fill up this space, but thereafter there wasn't a mutant which occupied the next region of the plate which contained antibiotic at the concentration of 1000 folds MIC. Why did this not happen is one of the main reasons for that is that going from 0 to 1000 needed many mutations. This transition of the bacteria being able to resist the concentration at MIC to increase that 1000-fold probably needed many mutations. So, many mutations were needed. Whereas, as we discussed very early in the course, that when we had two sequences, when we had a sequence space, I want you to remember and go back to the discussion where we said that if we have a sequence of length L, then 4 to the power L sequences are possible. And when we made this sequence space where each of these nodes represented one sequence, then we joined sequence I and J if they were connected, if the Hamming distance between I and J was equal to exactly 1. We never connected nodes which represented sequences which were separated by Hamming distance of more than 1. And that is important in this context, because why we said we only connected Hamming distance 1 was that if we have a population here, then in a division event, what might happen is that one of these individuals might acquire a mutation and the progeny belongs to this node. However, if we have another node K such that Hamming distance between J K is 1, it is not very realistic to expect that an individual which was at node I picked up a mutation and the progeny landed up at a node which was at a Hamming distance of 2 from it. And that is what we see here, that because an individual needed multiple mutations to increase its resistance from MIC to 1000 times MIC, those mutations could not have happened in one individual, as a result of which the population could not break through this frontier of 1000 times MIC. So that is one. The second thing is that this was an environmental stress. If we think of what we asked the bacteria to do here, this was a challenge that was too steep. If we in the original video that we saw, the challenge was broken down into smaller steps, where we went from 0 to 1 to 10 to 100 and eventually to 1000. So this was broken down into 4 steps, but when we remove those 4 intermediate steps and ask the population to come up come up with a solution to this environmental challenge that is posed in just one go, the population can't cope up with it. And if this was the environmental change that happened of 0 to 1000, it would just go extinct. And this is interesting in the context of what we are seeing today in an ecological context. The earth has been warmer in its history than it is today. The oceans have been more acidic than they are today, but the important parameter here is the rate at which the temperatures are changing and the rate at which ocean's pH is changing. Species cannot cope up with environmental change if it is too rapid. If the environmental change is slower, then species can adapt, they can acquire beneficial mutations and adapt to the stresses that are being posed. However, if the stress that is being posed is too harsh, then it is not possible for populations to come up with the genetic solutions that are necessary to adapt. As a result of which, because environmental changes are happening at a speed which have not at speeds which have not been seen previously, what is happening now is that the extinction rates of species today on our planet are 1000 times more than what are the background natural extinction rates present in ecology. So, that is an important illustration I feel that which comes out of this experiment also. The last point about this experiment is that when we talk of antibiotic resistance, the popular mode of resistance is something called horizontal gene transfer. So, we say that we have bacteria and this bacteria is susceptible to antibiotics. What that means is it has a very low MIC. And now if it were to acquire to become resistant, the more popular path that is thought to becoming resistance is via acquisition of genes from the environment. That if this is the genetic content of the individual, then maybe it will acquire foreign pieces of DNA and this piece of DNA will confer resistance. This has a certain gene on it and that gene's job is to tackle the action of the antibiotic that is present in the environment and confer resistance. And these are mobile genetic elements which can move from one individual to another. And typically these can move across from these can move across via different processes. They can be taken up from the environment. Phages, which are viruses that infect bacteria, can move pieces of DNA from one bacterial species to another, or you can have actual trade of genetic elements between bacterial species which are close to each other and have the right equipment to trade DNA strands with each other. So, resistance is commonly thought and easily spread through these type of mechanisms where I just acquire a foreign piece of DNA that I did not have previously, and this foreign piece of DNA that I have acquired confers resistance in me and I am able to survive the antibiotic. However, what we saw in the paper in the last video was that mutations can take place in the existing DNA of the organism that I did not acquire any foreign piece of DNA, mutations just happened in the genome that I am carrying already and these mutations facilitated increase of MIC up to 1000-fold. what that means is that we don't even need to invoke this horizontal gene transfer to explain increase in resistance. The genetic material present inside a cell is sufficient to acquire mutations and itself become resistant without acquisition of foreign pieces of DNA, which will of course aid this process, but this in itself can acquire mutations and become resistant. All right. So, that was the first study. Next, we are going to discuss perhaps what is the most famous evolution experiment in microbiology, which is called long-term. The experiment has a name which is called long-term evolution experiment. Or in short, it is referred to as LTEE. This experiment is so famous that in fact it has a Wikipedia page dedicated to it. So, if we just read through the Wikipedia page, this says long-term evolution experiment. LTEE is an ongoing study begun by Richard Lenski at California Irvine, and then the experiment was moved to Michigan State University when Lenski moved from Irvine to Michigan State University. And in the last couple of years since Lenski has retired, this is been taken over by Jeffrey Barrick at University of Texas Austin. And this was a study that was started more than 35 years ago in February of in February of 1988, and this is a little bit dated. This is 73,000, but as of now the experiment has crossed more than 80,000 generations have been processed in this experiment. So, in terms of volume and wealth of knowledge of what evolutionary processes can do from a lab experiment that we can gather, there is no other experiment which has told us more than this particular experiment. So, this has been going on since 1988, and to date it has processed more than 80,000 generations. This literally 100 papers have resulted from these studies. Some of them are just classical papers that we will discuss in this course, and this was started by a person called Richard Lenski. And much of the experiment actually took place at Michigan State University. Currently, the experiment is being led in Jeffrey Barrick's lab at Austin. Okay. So, while this is an experiment that was started roughly 35 years ago, the first microbial evolution experiment was done almost 100 years ago, 100 years before Lenski started this particular experiment. In that experiment, so this is the first microbial evolution experiment. This was started by somebody called William Dallinger. And what Dallinger was trying to do is the question that he was interested in was that suppose you have bacteria that can grow up to certain temperatures but not beyond that. Can you evolve these bacteria so that they evolve to be able to acquire this ability of growing at higher temperatures? And over an experiment that over the course of 7 to 8 years that this experiment lasted, Dallinger began this experiment by growing the bacteria at temperatures that were very low, so of the order of 20-25 degrees Celsius, and over 7-8 years he gradually raised the temperature of the media in which these bacteria were growing and was able to reach temperatures of the order of 70 degrees Celsius. Now, some people conjecture that we are not sure if there was no contamination. So, the original species had died down and some external contaminant had come out and was growing, and this is obviously the 19th century. So, techniques weren't sophisticated back then. But but assuming that there was no contamination and actually this adaptive process occurred, that bacteria which was not able to grow at 70 degrees was now growing at 70 degrees as a result of this experiment that lasted several years. Dallinger was also able to show a phenomena called trade-off in bacteria. What he saw, that the ancestor that he started with was able to grow at 25, but exhibited 0 growth at 70 degrees Celsius. The evolved strain, which was growing fine at 70 degrees Celsius, when he compared its growth at 25, the evolved strain was able to grow at 25, but not as well as the ancestor strain at 25 degrees Celsius. So, what Dallinger noted was that the ability to grow at 70 degrees Celsius came at a cost which reduced the fitness of this bacteria at 25 degrees Celsius. So, it told him this idea of trade-offs, that you can't be very good at growing at both very low and very high temperatures. As you become better to grow at higher temperatures, your ability to grow at lower temperatures is compromised. And this was the sort of the first demonstration of the phenomena called trade-offs. The ancestor, so let us say this is 30 degrees Celsius, this is 70 degrees Celsius. So, ancestor did high growth rates, exhibited high growth rates at 30 and 0 growth at 70. It was not able to grow at this elevated temperature. On the other hand, the evolved strain exhibited growth at 70 degrees Celsius, but the growth at 30 degrees Celsius was intermediate, was not as high as the ancestor. So, what this tells us is that in order to become better at 70, so in 70 the performance improved after this evolution experiment, but that came at a cost of decreased performance at 30. Now, are these trade-offs necessary or is it just the fact that you were not growing at 30 degrees Celsius, you had not seen temperature of 30 degrees Celsius for several years and hence there was no need to maintain the ability to grow fast at 30 degrees Celsius, are the kind of questions that we are interested in as a field and studying these trade-offs which are encoded by cellular logic. The experiment I think ended in a bit of a disaster because I think the equipment just burst. So, almost 100 years on from Dallinger's experiment, Lenski started the following experiment. So, LTEE has 12 identical lines with some minor differences. There are groups of 6 each, but we will not consider that difference here. It is not relevant for the discussions that we will be having. And starting with an ancestor E. coli, so this is ancestor E. coli, it has its genome. This ancestor was placed in a flask which contained low amounts of glucose as the carbon source. And this ancestor was placed and growth was allowed to happen in this flask for a period of 24 hours, after which the flask would have lots of individuals present in them. and then 1:100 dilution would take place to a fresh flask which contained the same media as the one that was started with. And that is pretty much the entire experiment, and this process has been continued on since Feb of 1988. Except for the fact that when this experiment was started, Lenski did not start only one line, but he started this experiment with 12 identical lines. So, some of the ancestral cells were added to another flask which had an identical composition as the first flask, and an identical treatment was meted out to this as well. So, this we will call this. So, these independent repeats, identical independent repeats are called lines. So, this is line number 1, line number 2, and there are, as we listed here, that there are 12 identical lines which have been going on since. Each one of these lines has been evolved for 80,000 generations. And the idea is to understand what sort of phenotypic and genetic changes. So, let us do it on the next slide. So, what are some of the questions of interest here? That what are the sort of phenotypic and genetic changes that take place in these populations as they are evolving for such a long period of time. Between the 12 lines, is the response parallel? Well, is the response identical or distinct? What that means is that, is it that line number 1, for instance, exhibited many beneficial mutations very quickly, whereas line number 2 did not do anything, and so on and so forth. Are the dynamics of the adaptive process same or very different? Are the kind of mutations that these lines are acquiring same or different in these populations? Are some of the kind of questions that this work was started with. Last point to note here is that when this experiment was started, the carbon source in this flask is glucose. However, this environment, this liquid media that the experiment was performed in, also contained quantities of citrate. Citrate as a carbon source in the presence of oxygen cannot be used by E. coli for growth. So, E. coli in oxic conditions, these flasks were shaken well, so there was plenty of oxygen around. In oxic conditions, cannot be used as a carbon source. So, what that means here is that you have this resource available which is being used and competed for in the population. However, you have this another resource present in the population which is not being able to be used because of an environmental condition. If the environmental conditions were different, and suppose if oxygen was not present, then E. coli can use citrate as a carbon source. However, in the context in which this experiment is being done, citrate is not available as a carbon source. So, you have this environment where there is this additional resource present, but just out of reach of the population. And what would happen in that case? So, obviously this experiment is a long story of 80,000 generations. My guess is like 100 papers have come from these type of studies. And we will start with, it is impossible to cover everything, but we will start with a few key results that we will discuss as part of this course. About 10 years into the experiment, late 90s, is I think when Lenski himself says that there was nothing really exciting going on in the experiment, and he had thoughts about shutting the experiment down. But close to the turn of century, something interesting happened, something remarkable happened, which led to a very exciting discovery of how evolutionary change can take place and how we can study them through experiments done in lab. And since then, this experiment has now become an institution, and there is no experiment that has gone on for longer and has told us more about the molecular basis of adaptive responses of populations from the context of bacteria in this case. And we will start our discussion of some of the key results from this LTEE study from the next video onwards.